Hello and welcome all in this video. So we are going to discuss about the life cycle of Entamoeba histolytica. So the Entamoeba histolytica is one of the well-known parasite, and in this video we are going to discuss about the life cycle of this particular Entamoeba histolytica. So this organism Entamoeba histolytica is a monogenetic organism. Monogenetic organism, that is the meaning of monogenetic, that is this organism required only one host to complete their life cycle. This organism required to complete only one host to complete their life cycle, so called as monogenetic. That is, this primary host or the main host of this organism is the human being or the man. So this Entamoeba is a monogenetic organism required only one host to complete their life cycle. But we can also found there are some reservoir hosts like pigs, dogs, rats, or rabbits. This organism is also found in the pigs, in the body of the pigs, dogs, rats, and rabbits. These are supposed to be the reservoir host of this organism. So the life history, when we see the life cycle of this organism or the life history of this organism, they can follow the two-way life cycle. In that, there is a first step, first type of life cycle we can found in this organism or followed by this organism is the binary fission. There is a single organism, then the single organism divided into two organisms. That process is called as a binary fission. So whenever this binary fission takes place, the organisms can be divided into two individuals. So let us see in the next slide about this binary fission. So here is the diagram of the binary fission. So first of all, this organism that is called as trophozoite. The organism which is called as trophozoite, this trophozoite organism divided into two individuals. Suppose it's a trophozoite and this form is a precystic or minuta form. Precystic or trophozoite, precystic or minuta form converted into the trophozoite form, and this trophozoite form is a infective stage of this organism. So these trophozoites multiply asexually by binary fission. That is, from one organism, two organisms are formed. So at the time of binary fission, first of all, and this process of the binary fission takes place in the wall of the large intestine of the human being. First of all, the nucleus is divided. First of all, nucleus is divided. Nucleus is divided into two nuclei. That is, this nucleus undergoes by dividation undergoes with the help of mitosis. The nucleus undergoes mitosis without the disappearance of the nuclear membrane, and after the dividation of the nucleus, then cytoplasm is also divided. That is, cytokinesis takes place. That is the division of the cytoplasm. First of all, the nucleus is divided, and the nucleus is divided, and this nucleus is then dividation of the nucleus is then followed by the cytokinesis. At that time, the division of the cytoplasm takes place. As a result, two daughter organisms are formed. Two daughter organisms are formed from a single organism, and that process is called as a binary fission. Then these organisms grow rapidly in size and feeding on bacteria and host tissues and other elements, and again multiply by the binary fission. So this can be takes place many times. From single organisms, two organisms are formed, and again then this precystic or minuta form is formed from this trophozoite form converted into binary fission into the binary into the precystic or minuta form. This is the first type of or a type of binary fission which takes place in the life cycle of the Entamoeba histolytica. Then again, there are also multiple fission takes place. That is, from one organism, about 8 organisms are formed in that process. That is called as a multiple fission. This multiple fission is also found in this organism. So again, Suppose this is a minuta or pre-cystic form, then they convert into the trophozoite form, then uh, then form pre-cystic stage. And in this pre-cystic, at the time of pre-cystic stage or minuta stage, the pre-cystic or minuta form at the time around this particular organism, a layer is formed, that is cyst formation takes place around this organism, and the cyst formation takes place, and the pre-cystic or minuta forms. The process of formation of cyst is called as encystment. So encystment takes place in this stage, and this can be takes place in the lumen of the intestine and not in the tissues, but this process takes place in the intestine. So around that particular organism, that the organism round up and secrete a thin and tough, flexible cyst wall around that organism. Around the organism, the cyst wall is formed, and the cyst at this stage is uninucleate. At this time, this cyst is uninucleate. That is, we can found in this organism, the cyst is uninucleate. There is having only one nucleus. So this cyst is uninucleate, and the single nucleus then undergoes multiple division or mitosis division to form two nuclei. So again, the single cyst is then converted into the binucleate cyst. Binucleate cyst, in this stage, two nucleus are formed. In the pre-cystic stage, there is having one nucleus. Around then this one single nucleus is divided into two nucleus and forming binucleate cyst. So in this diagram, we can find there are having two nucleus. Then that is uninuclear stage, and followed by the binuclear stage, in which there is having two nucleus. Then again second mitosis takes place, and these two nucleus again divided into and forming four nucleus, and that stage is called as quadranuclear cyst, or then also called as tetranuclear cyst. So binuclear cyst then converted into the tetranuclear or quadranuclear cyst. Tetranuclear or quadranuclear cyst. Then this tetranuclear cyst constitutes the transmission or infective stage. This quadranuclear or tetranuclear cyst is the infective stage. Then again there is no further development do not take place, but this cyst then pass then this cyst then pass out of the host in the fecal matter or feces of the host. Then cyst is then passes by this organism. So afterward, after development of the quadranuclear stage, four nucleus are formed. Then this cyst, quadranuclear cyst, then comes out of the cyst, and that process called excistation. Then excistation takes place. That is, the organisms or quadranucleate organism then comes out of the cyst. So they can remove the cyst or the envelope of the cyst, and that is called as excistation. Afterward, this excistation stage or this organism then passes out of the body of the host, and they such type of cyst then alive for about 10 days in the moist stool. In the moist stool or in the waste material of the human being, they can remain about 10 days. And afterward, and suppose the temperature rises above 50 degrees Celsius, then they can be destroyed. But they can remain for 10 days in the moist stool. Then this is the infective stage, or this stage is called as infective stage, or this stage is a transmitted stage or infective, and this infection depends upon the intake of food. This stage is called as infective stage, and this can be depend upon the intake of food or water, and that food must be contaminated. That is, suppose this food is contaminated, then contaminated with this particular type of cyst, then that cyst can enter into the another host. So infection depends upon the intake of food or water contaminated with the fecal matter containing tetranuclear cyst. The food handlers like sorry, food handlers like cooks, sweet meat sellers, or hawkers can transmit such type of quadrinucleate cyst. And untreated human waste material which are also transmit such type of cyst into the another host, into the new host. So again, it is a common source of infection. Houseflies and housefly and cockroach also can transmit or carry this quadrinuclear cyst on their legs or in their intestine and transfer them to the unprotected food materials. So this can be with the help of the handlers, food handlers or cockroaches or houseflies can transmit this quadrinuclear cyst into the new host. And afterward, after the excision, then in the new host, the ingested cyst passes down the alimentary canal and reaches into the small intestine. The cyst wall protects them from the action of the host gastric juice, and during their passage through the stomach, after 5 or 6 hours after the entry into the body of the body into the new host, after 5 to 6 hours, this excision process takes place. That is, this paranuclear cyst comes out of the cyst wall and excision takes place as the cyst wall is digested by the trypsin in the small intestine, and this again forming amoebic or metacyst. Then this cyst afterward, after excision, then many excystic amoeba or the metacysts are formed. Excystic amoeba or metacysts are formed, and further this metacystic development takes place by, that is, from a single organism. There are having 8 such type of metacysts are formed. These metacysts are uninucleate organisms. So from a single propozoite, about 8 small uninucleate metacystic propozoites are formed, and this is one of the type of multiple division which takes place in the life cycle of the Entamoeba histolytica. So these are two stages about the development or the life cycle of this Entamoeba histolytica. So again, in the next video, we will discuss about the disease which are caused due to the Entamoeba histolytica and how we can control over that disease. So if you like this video, then share with your friends and subscribe the channel. Thank you.